Bismillahirrahmanirrahim. I welcome all my students to today's lecture of continuity of endocrinology. And today we are going to learn about the feedback control mechanism of hormone secretion. Now, as you all know, that there is a control system of hormones in our body, and which is most likely and up to some extent be controlled by the feedback mechanisms. Now, the feedback prevents overactivity of the hormone systems. The plasma concentrations of many hormones fluctuate in response to various stimuli that occur throughout the day. In most instances, this control is exerted via negative feedback mechanism to ensure the proper level of hormone activity at the target tissue, which is composed of the trigger cells. Trigger cells are those cells which have the receptors specific for that particular hormone in them. Um, Now, after a stimulus causes release of the hormone, conditions or products resulting from the action of the hormone tends to suppress its further release. In other words, the hormone or one of its products has a negative feedback effect to prevent over secretion of that particular hormone or overactivity at the target tissue in order to maintain the homeostasis. Feedback regulation of hormones can also occur at the levels of protein formation, which includes the gene transcription, gene translation. The steps definitely involved in the synthesis of hormones, the steps involved in processing hormones or releasing the stored hormones. Now, the surges of hormones can occur with positive feedback mechanism as well. Now, the positive feedback mechanism occurs when the biological action of the hormone causes additional secretion of the hormone. One of the beautiful example is the surge of luteinizing hormone, which occurs as a result of the stimulatory effect of estrogen on the anterior pituitary before ovulation. The secreted luteinizing hormone then acts on the ovaries to stimulate additional secretion of estrogen, which in turn causes more secretion of luteinizing hormone. Luteinizing hormone reaches an appropriate concentration, and typical negative feedback control of hormone secretion is then exerted. Cyclical variations occur in hormone release. The negative and positive feedback control of hormone secretion are periodic variations in hormone release that are influenced by seasonal changes, various stages of development and aging, the diurnal daily cycle, and sleep. For example, the secretion of growth hormone is markedly increased during the early period of sleep but is reduced during the later stages of sleep. In many cases, these cyclical variations in the hormone secretion are due to the changes in activity of neural pathways which are involved in controlling the hormone release. Now, when these hormones they are being released in the blood, there should be some transport method of the hormones in the blood. Some of them, as you all know, they are water soluble. Some of them are steroid and thyroid hormone, which are not water soluble. So the water soluble hormones like catecholamines, peptides, they are dissolved in plasma and are being transported from their sites of synthesis to target tissues, where they diffuse out of the capillaries into the interstitial fluid and ultimately to target cells. Steroid and thyroid hormones, in contrast, circulate in the blood mainly bound to plasma proteins. Usually, less than 10% of steroid or thyroid hormones in the plasma exist free in solution. For example, more than 99% of the thyroxine in the blood is bound to plasma proteins. Protein bound hormones cannot easily diffuse across the capillaries and gain access to their target cells and are therefore biologically inactive until they dissociate from plasma proteins. The relatively large amounts of hormones bound to proteins serve as reservoirs, replenishing the concentration of free hormones when they are bound to target receptors or lost from the circulation. Binding of hormones to plasma proteins greatly slows their clearance from the plasma. Clearance of hormones from the blood. Metabolic clearance rate is equal to rate of disappearance of the hormone from plasma over the concentration of hormones. Now, the two factors can increase or decrease the concentration of a hormone in blood. One of these is the rate of secretion into the blood, and the other one is rate of removal from the blood. So, this is the metabolic clearance rate and is expressed in terms of the number of milliliters of plasma cleared of the hormone per minute. The usual procedure for making this measurement is following that a purified solution of hormone to be measured is tagged with a radioactive substance, then the radioactive hormone is infused at a constant rate into bloodstream, and later the radioactive concentration in the plasma becomes steady, and at this time the rate of disappearance of the radioactive hormone from the plasma equals the rate at which it is infused, which gives one the rate of disappearance. At the same time, the plasma concentration of the radioactive hormone is measured using a standard radioactive counting procedure, and hormones are cleared from the plasma in several ways, including metabolic destruction by the tissues, binding with the tissues, excretion by the liver into the bile, excretion by the kidneys into the urine. Now, for certain hormones, a decreased metabolic clearance rate may cause an excessively high concentration of the hormone in the circulating body fluids. For instance, this occurs for several of the steroid hormones when the liver is diseased because these hormones are conjugated mainly in the liver and then cleared into the bile. Hormones are sometimes degraded at their target cells by enzymatic processes which cause endocytosis of the cell membrane hormone receptor complex. The hormone is then metabolized in the cell and the receptors are usually recycled back to the cell membrane. Most of the peptide hormones and catecholamines are water soluble and they circulate freely in the blood. They are usually degraded by enzymes in the blood and tissues and rapidly excreted by the kidneys and liver, thus remaining in the blood for only a short time. Hormones that are bound to plasma proteins are cleared from the blood at much slower rates and may remain in the circulation for several hours or even days. The half life of adrenal steroids in the circulation, for example, ranges between 20 and 100 minutes, whereas the half life of protein bound thyroid hormones may be as long as 1 to 6 days. Now, one of the very frequently and most important topic which is being asked from the hormone is the mechanism of action of hormones, and we should be knowing about this in detail. What is the mechanism of action of hormones? What are hormone receptors? How are they being activated? Now, always remember that the first step of hormones action is to bind to specific receptors at the target cell. Cells that lack receptors for the hormones do not respond. Receptors for some hormones are located on the target cell membrane, whereas the other hormone receptors are located in the cytoplasm or the nucleus. When the hormone combines with its receptor, this usually initiates a cascade of reactions in the cell, with each stage becoming more powerfully activated so that even small concentration of the hormone can have a large effect. Hormonal receptors are large proteins, and each cell that is to be stimulated usually has some 2000 to 100,000 receptors. Also, each receptor is usually highly specific for a single hormone, and this determines the type of hormone that will act on a particular tissue. Now, we should be knowing about the location of the different hormone receptors, which are as follows. They are in or on the surface of cell membrane, they are in the cell cytoplasm, or they are in the cell nucleus. Now, in or on the surface of the cell membrane, the membrane receptors, they are specific, mostly for the protein, peptide, and catecholamine hormones. Now, the primary receptors for different steroid hormones, they are found mainly in the cytoplasm. And the receptors for thyroid hormones, they are found over there in the nucleus and are believed to be located in direct association with one or more of the chromosome. So always keep this thing in mind that the receptors for the protein, peptide, catechol hormones, they are over there on the surface of the cell membrane. The receptors for the steroid hormones, they are over there in the cytoplasm, and the receptors for the thyroid hormones, they are over there in the nucleus, in close association with one or more of the chromosomes. Now, the important thing is the number and sensitivity of hormone receptors, and they are always regulated. The number in the target cell usually does not remain constant daily or from day to day, even from minute to minute, depending upon the activity of that particular cell. It varies. The receptor protein themselves, they're often inactivated or destroyed during the course of their function, and at the other end times they are reactivated or new ones are manufactured by the protein manufacturing mechanism of the cell. For instance, increased hormone concentration and increased binding with its target cell receptors sometimes cause the number of active receptors to decrease. This downregulation can occur as a result of inactivation of some of the receptor molecules, inactivation of some of the intracellular protein signaling molecules, temporary sequestration of the receptor to inside of the cell, so making the site of action to interact with the cell membrane away. So this is the downregulation of the receptors, making less availability of the receptors for the attachment of the hormone. Destruction of receptors by lysosomes after being internalization, decreased production of the receptors. In each case, receptor downregulation decreases the target tissue's responsiveness to hormone. At the same time, there is upregulation of receptors for hormones. That is, by stimulating hormone induces greater than normal formation of receptor. Intracellular signaling molecules by the protein manufacturing machinery of the target cell, or the greater availability of the receptor for interaction with the hormone. Intracellular signaling after hormone receptor activation. A receptor affects its target tissues by first forming a hormone receptor complex. This is the first step, and this is going to alter the function of the receptor itself. And the activated receptor initiates the hormonal effects. So there is presence of ion channel linked receptors, G protein linked hormone receptors. So we are going to learn about all this detail inshallah tomorrow, and you should be learning all the details which we have done today. What is the what are the different characteristics of hormones? What is the mechanism of their feedback control mechanism? What are the sites of receptors for different hormones? What is the upregulation? What is the downregulation of receptor? So this is all about today's lecture. So stay connected, stay blessed. Allah Hafiz.